Hello, I'm Marc Schallenberg from the University of Otago, and I'm going to be talking about critical step 1.3.2, food web biomanipulation techniques to enhance ecological processes. Now, I'm going to be talking about classical food web biomanipulation, which has been around in Europe and North America for a long time, but it's never really been trialed in New Zealand. And a couple of papers that have been published by our group here at the University of Otago suggest that this biomanipulation technique could be useful in the New Zealand context, particularly since we have a new invasive species of Daphnia, the water flea, that exists in our lakes now. And I'll explain this a bit more as we go. So this critical step involves myself, Dave Kelly from the Cawthron Institute, Pete Verberg from NIWA, Kevin Collier from University of Waikato, Ian Duggan and Brendan Hicks also from the University of Waikato. And we have half a PhD student working on this project with some operational funding. The PhD student is shared with critical step 1.2.3, which is about food web responses and biodiversity. There are a number of action partners that we'll be working with in this program, including the Otago Regional Council, Otago Fish and Game, and Friends of Lake Hayes around the work that we'll be doing in Lakes Hayes and Johnson, two eutrophic lakes in central Otago. Potentially working on Lake Naroto as well with the assistance of Environment Waikato. Lake Panui in Greater Wellington region is a small isolated lake that also contains perch that would be very interesting to look at as a comparison lake to Lakes Hayes and Johnson. And potentially Lake George or Rawira, which is in the Southland region. And that lake is a lake that's being restored by the Oraka Runaka. And so far they've done a great job of restoring the wetlands around the lake, but I think from preliminary discussions they would be very interested in potentially removing perch from this lake, which could be very interesting from the perspective of the work in this program. And I hope that will be clearer by the end of this presentation. So this This all fits into kind of conceptual framework of how lakes respond to pressures. And this is just a kind of theoretical response and pressure diagram for a lake. And the response here would be algae, where we have healthy to degraded, and the pressure here would be nitrogen loading or nutrient loading, where we go from low to high. And what generally happens in shallow lakes or lakes that have complex food webs is that with low levels of nutrient loading, the lake is able to absorb quite a lot of this nutrient without any major changes in the key values, say the algal biomass. And this is due to there being stabilizing feedbacks in the lakes. And grazing is one of these potential stabilizing feedbacks, zooplankton grazing. But the idea is that when you eventually put in so much nutrient that it saturates these feedbacks and you get an abrupt change to a degraded state, and this degraded state has its own feedbacks, its own inertia, which make it very difficult to restore back to the healthy state. And the differences in trajectories between degradation and restoration here is called hysteresis. Hysteresis is really the distance between these two thresholds, and it relates to a nutrient load. And the idea is that these stabilizing feedbacks create conditions where you really need to bring the nutrient loading down to extremely low levels to promote a switch back to the healthy state. And this is very costly. And the idea behind this food waste biomanipulation is that it could potentially reduce this hysteresis so that we, during the recovery phase, we would get a better, quicker response to nutrient reduction than we would without biomanipulation. The biomanipulation would break some of these stabilizing feedbacks and allow the system to flip back, hopefully near the same threshold that it flipped in the first place in the degradation phase. And this green line here shows what I would call the intervention gain. So by using biomanipulation, you're using an intervention, and the gain here is the difference between the old hysteresis and the new hysteresis, and this is equivalent to a nutrient load that doesn't need to be achieved anymore, a reduction in nutrient load that doesn't need to be achieved anymore to restore the lake. And so this is a big cost saving and a big time saving. It can help basically kick the lake back into a healthy state much more quickly. And this is the idea behind biomanipulation, is that ideally it would do this in a lake that you're trying to restore, which might have some inertia to restoration. Lake Hayes is a lake that we have a great deal of interest in with regard to biomanipulation as a potential restoration tool. And here's some data showing the Secchi depth going back from the 1940s right through to the present day. And around here in the late 1960s, early 1970s, the lake began to degrade, and we can see that some of these summer Secchi depths are quite low, below 3 meters of water clarity. And the lake was considered to be quite degraded, and it had cyanobacterial blooms, for example, and locals were not very happy with the degraded state of the lake. Unfortunately, it got worse in the mid-2000s when Serratium, a dinoflagellate, became the dominant alga in the lake, and this created, it seemed to be an enhanced phytoplankton bloom and really drew down the Secchi depths to extremely low levels, between 1 and 2 in some cases meters of water clarity. And so this was quite a desperate situation since about the mid-2000s, and there's been a lot of thought put to what could be done to ameliorate this. Now, interestingly, within this time period when Serratium was the dominant alga and causing lots of algal bloom problems in summers, there were a couple of years where the lake achieved unprecedented water clarity. Now, this year here with the red dots is year 2010. And this one is just this past year. These are data from 2017, the summer of 2017. So we had extremely clear water and virtually no Serratium in these 2 summers. And yet, between those summers, we had extremely bad, historically bad water clarity. So this lake seems to be going through some kind of a bifurcation, some kind of a flipping back and forth between degraded water and then suddenly clear water and then going back to degraded water and then clear water again. And of course we're very interested in what could be the mechanisms behind this, what could be the drivers, and we think that biomanipulation and food web interactions could be something to look at in this particular lake. One of the reasons we think that is because when we look at the densities of Daphnia, the water flea, the main grazer of algae in these lakes, if we look at the summer Daphnia densities, we see that in 2010, the year where there was unprecedented clear water, we had Daphnia persisting in quite high numbers throughout the summer. In 1995, we also had some Daphnia persisting, but at much lower levels, and these were Daphnia carinata, the native Daphnia. But between 1995 and 2010, Daphnia pulex invaded the lake, and Daphnia pulex does reach higher densities and does seem to be a much stronger grazer on algae. In 1970 and 71, when we had very bad cyanobacterial blooms, there were no Daphnia that persisted during the summer. So we're wondering if Daphnia persistence during the summer might have something to do with these clear water summers, and this has been reinforced because just this past summer there was another clear water summer, and in that summer the Daphnia persisted again throughout the summer months. So Daphnia might be having some kind of interesting interaction with the algae in this lake, and the reason I say it's an interesting interaction is because these Serratium are too big for the Daphnia to graze directly. So whatever interaction there might be going on between Daphnia and Serratium, it's not a direct grazing interaction, it's something indirect that seems to be occurring. I have a student, an MSc student, Helen Trotter, who's been also interested in biomanipulation and some of the questions, the fundamental questions around whether biomanipulation could work in these lakes. And so she's been collecting some of the fundamental information in Lake Hayes that could help us understand whether biomanipulation would work, and if we were to apply it, how best we could apply it in the most cost-effective way. And so she's been doing things like monitoring the Daphnia densities from September 2015 through to August 2016, but also the Secchi depth, the water clarity of the lake, and also been looking at different fish species in the lake, in particular focusing on the perch and their spawning and when the juvenile perch are most abundant. And what she's shown here in 2015 is that while they spawn in September and October, they reached high juvenile abundance in December and January, and as they're increasing, the Daphnia densities crashed in the lake. And there were no Daphnia in January and February, and very low levels in March, April, and May. And concomitant with that is this decrease in the water clarity. Although when Daphnia started to bounce back, water clarity improved somewhat as well, to the point where in August we had healthy Daphnia numbers again, and also very high water clarity. So this is starting to provide some interesting insights into the timing of this key interaction, potential key interaction between perch and Daphnia and algae. And it's looking like this period of perch spawning and then the juvenile hatch, basically the hatch of the spawn, and the juvenile perch abundance in December and January being key times for this interaction. So we've got and a couple of hypotheses for Lake Hayes, and we wonder if these hypotheses can be confirmed by further study and whether they could be generalized to other lakes that contain perch and have algal bloom problems. Basically in Lake Hayes we have two piscivores. We have large perch and we have brown trout, which feed on the small perch. The small perch feed on the Daphnia. The Daphnia don't feed on the algae, but they do have some sort of negative interaction which we don't understand yet. But there are some other complicating factors in this lake. We have a few coaro, not very many coaro in the lake, which most likely also feed on Daphnia. And we have bullies, and the larval bullies may also feed on Daphnia as well. So it may not be as simple as just focusing on the perch and the young of the year perch. Other things could be going on as well. And so we're very interested in trying to tease apart these trophic interactions to try and understand which are going to be the key ones that drive Daphnia and consequently algae in the system. So our overall hypothesis that we have, just based on observations in the last, I don't know, 8 years or so, is that perch recruitment has something to do with the dramatic improvements in water clarity, and that we wonder whether perch recruitment was bad in the years 2010 and 2017, resulting in dramatic improvement in water clarity through allowing the Daphnia to persist over the summer. We can kind of rephrase this in a way that provides some really interesting and testable hypotheses for our research. And that is that basically perch presence increases phytoplankton biomass via the young of the year perch, which prey on Daphnia pulex. And that increases hysteresis and inhibits restoration of the lake. Lakes with perch and Daphnia pulex may benefit from pelagic food web manipulation, and that's the more generalized form of the hypothesis that we're testing in Lake Hayes, potentially Lake Johnson, potentially Lake Panui, potentially Lake George. Why should stakeholders care about biomanipulation to remove perch? Well, the removal of perch could reduce the hysteresis and make restoration from eutrophication more feasible and cost-effective. The removal of perch restores more nativeness to lake food webs basically by reducing predation on native fish like kawaro and bullies, and by potentially encouraging eel restocking, because we think that eels might be an important factor in what we see going on in these lakes, and I'll talk a little bit about that in the next slide. So there are a number of potential biomanipulation approaches that we've been thinking of, kind of in anticipation of the results of our studies. If we were to find these strong trophic interactions, which would suggest improvement in water quality through somehow affecting the young of the year perch, we could look at things like perch removal, netting of the perch. We could look at things like trying to rear sterile perch to larger sizes where they're piscivorous, and then stocking the lake with piscivorous perch, which are sterile. We could try to potentially encourage Fish and Game to stock trout. The trout in the lake are not stocked, they're self-sustaining population, but potentially increasing trout stocks in the lake could help reduce young perch densities. We could potentially use perch spawning substrates, which we could put in the lake and then remove after spawning before hatching of the eggs, and thereby trying to cause a perch recruitment failure, or at least a poor year for perch recruitment, which could potentially then allow the Daphnia to survive over summer. We could encourage perch fishing, or we could encourage the stocking of eels, which potentially would feed on the young perch. And eels have actually been locked out of this system since the Roxburgh Dam was put in the 1950s, because they cannot get over the dam. And so there is a trap and transfer program at the moment, but they do not move the eels above the Clyde Dam. They only move them over the Roxburgh Dam. So there's a potential there for maybe restocking eels into Lake Hayes with the help of Contact Energy, who are doing this trap and transfer. That's definitely something we're interested in looking at as part of this, the potential biomanipulation. So we have a work plan, which at this stage is a preliminary work plan. It involves this PhD student, appointing the student, which we did. The student begins studies. Well, that didn't happen in the first quarter. It happened in the second quarter of this year. So we're a bit slow there, and this was because the student had some major delays in getting their student visa, unfortunately. But some of the ideas for doing work include conducting enclosure experiments to measure the grazing pressure of Daphnia, to see if it could potentially graze on serratium, or to look at other potential interactions between Daphnia and serratium, by which Daphnia could suppress serratium. We could look at historical Daphnia dynamics in relation to some key events in the lake, which we know the dates of. For example, the introduction of perch to the lake, the introduction of trout to the lake, the first years when the lake became quite eutrophic, having major cyanobacterial blooms. What went on with Daphnia dynamics in relation to these key events in Lakes Hayes and Lake Johnson. Lake Johnson is a neighboring lake which also has perch and tends to go through the same sorts of dynamics as Lake Hayes does. We could take these sediment cores and date the cores and look at Daphnia remains in the core and reconstruct Daphnia population dynamics in relation to the different historical events that I just mentioned, which could provide insights into how Daphnia and fish interact and the algal blooms interact as well. We could use all this food web information that's being developed and collected to develop a food web model, which is really something that we will need to do to be able to run scenarios on how we could potentially manipulate the food web and whether or not we would expect significant improvements in water quality for different types of manipulation. So it's a scenario running tool. And we could do some larger enclosure experiments looking in more detail at the interactions between perch and Daphnia in these lakes. All of this would eventually lead towards some kind of feasibility analysis of biomanipulation and whether it would work in the lakes that we are studying, with a view to potentially carrying out a whole lake biomanipulation study towards the end of this research program. So the PhD student that we appointed is Samiula Khan, who did an MSc under Martin Kainz at the University of Vienna. And he specializes in food web science using biomarkers, in particular fatty acids, essential fatty acids, to determine trophic linkages in food web. So he's really well placed to do this food web work in our lakes. He's a student leader in the Global Glion project, so he has a very good international profile through that. And from my interactions with Sammy and from reports we've heard from people who've worked with Sammy, he's a really excellent student, and we expect some really great work to be done by Sammy. He's going to be co-supervised by myself, Kevin Collier at Waikato, and Jerry Kloss, who's the head of department in the zoology department at Otago University. And Sammy will work on two of the critical steps, 1.3.2 and 1.2.3, so biomanipulation and food webs and biodiversity. Unfortunately, we had some quite major delays in getting Sammy over here through delays with getting his visa, but that's now been sorted and Sammy's here, and he's been here for about a month, and we're working on his proposal, and that's coming together really nicely. So we've been a bit late, but we think we're underway now. Progress to date? Well, there's been, since 2015, since Helen Trotter began her work, there's been collections of zooplankton, chlorophyll A, nutrients from Lakes Hayes and Johnson through work that Helen and Otago Fish and Game and Otago Regional Council have done. So there's almost 2 years worth of data now for Lake Hayes and 1 year for Lake Johnson. There's been a collection of fish samples from Hayes and Johnson by Helen Trotter for a 1 year period. And there's an analysis of food web samples and data going on at the moment, and that's Helen's work that she's trying to get finished up so she can finish up her master's, which we expect to be done by the end of the year. So I've showed you some of her preliminary results, which show the timing of the perch spawning and the young of the year perch coming online in Lake Hayes and how that affected Daphnia. So yeah, we hope to follow this up with this year's data, which have also been a clear water year, and we know that, again, Daphnia persisted in the lake over this summer, so this will be an important year to kind of confirm the results from 2010, and luckily we've been getting data right through this time period for this year as well. So we have two nice years, one year where we had severe algal blooms and no Daphnia, and another year where we had clear water and Daphnia.